Good morning, everyone. Welcome to the seminar of the Biotechnology Center here from the Federal University of Rio Grande do Sul. Today we have the pleasure of having Dr. Carol Thiele from NIH, and I'm today also with Dr. Rafael Hessler from our university that is going to introduce Carol for us, and I have the pleasure to have you here. Rafael, please, the microphone is yours for doing the presentation and conducting the seminar. Thank you.
Hey, thank you, Felipe. Good morning, everyone. Bom dia. So today's seminar is part of the regular Center for Biotechnology seminar series, but it's also co-promoted by our recently launched National Institute of Childhood Cancer Biology and Pediatric Oncology, which is a project funded by the Brazilian National Research Council, CNPq. So it's a co-promotion of the Center for Biotechnology and our National Institute. So we're live on YouTube. Any of you who wants to ask questions during the talk can send them in the YouTube chat. If you want, feel free to ask questions in Portuguese. There's no problem, we can translate them for you. And as Felipe said, we're very pleased to welcome Dr. Carol J. Thiele as our invited speaker this morning. I hope to be pronouncing her last name correctly. Dr. Thiele holds a bachelor's degree in biology from the University of California, San Diego, and a PhD degree in microbiology and immunology from the University of California, Los Angeles. And she had postdoctoral training at the National Cancer Institute, NCI, at the National Institutes of Health, NIH, in Bethesda, Maryland, where she still is. She's currently a senior researcher and deputy chief of the Pediatric Oncology Branch and deputy director of the Center for Cancer Research at the National Cancer Institute. She's one of the world leaders in the field of childhood cancer biology. Her work has provided the basis for the introduction of retinoid therapy for patients with neuroblastoma. She also described new mechanisms of resistance to chemotherapy and how to overcome this resistance in neuroblastoma. And more recently, in the past years, she has focused mainly on the role of the regulation of embryonic development and epigenetic mechanisms in pediatric tumors. Carol has interacted with our Center for Biotechnology before. She has served as a co-supervisor of a PhD thesis with us in our graduate program in cellular molecular biology. She was the co-supervisor of Dr. Barbara Kunzler, who's certainly watching us now. And she's been a generous supporter and collaborator of our work here in Porto Alegre. She kindly serves as an international advisor and collaborator in several of our research grants. And she's a co-author of some of our papers that focus on epigenetics of pediatric cancer. So I'm very happy to welcome you here, Carol, this morning. Welcome. Thank you so much for taking the time to be with us. And I'm sure this will be a fantastic lecture.
Thank you very much, Rafael. It's really a pleasure to be able to talk to you all today. Neuroblastoma is my passion, and I think we've made a lot of progress, but there's certainly a lot to be done. So today I'm going to talk about how the oncogenic events dysregulate developmental pathways to promote tumorigenesis. Neuroblastoma really accounts for only about 7% of all childhood cancers, and in the US, that's about 800 newly diagnosed cases per year. Yet they comprise a disproportionate number of deaths due to cancer in children. And one of the things that always fascinated me is that it is a tumor of the peripheral nervous system, a tumor of neural crest origin. And, you know, the neural crest is really a fascinating structure. It's a transient structure that arises during the closure of the neural tube. And these cells migrate ventrally and laterally and give rise to a number of different tissue types. I mean, the plasticity of these cells is truly amazing. They become melanocytes. The medullary cells of the adrenal gland, they contribute to the craniofacial bony structures, parts of the vasculature system. So that really contributes to their heterogeneity. So the other fascinating things about neuroblastoma that led us to believe that there were alterations in development is this very interesting presentation of two children with neuroblastoma. Here you have an infant with this, what we call blueberry muffin presentation of nodules of neuroblastoma over their body, while you have another slightly older child who presents with a distended belly. When the pathologists looked at these tumors, they were really quite surprised because the tumors from this child were really fairly well differentiated, either called ganglioneuroma or ganglioneuroblastoma, while the tumor from this child here was a monomorphic sheet of small round blue cell tumors and frequently identified by what the pathological structure here called a Homer-Wright rosette. But what was interesting was these tumors, the ganglioneuroma and ganglioneuroblastoma, that had a much better prognosis, and it was this child here who really had a poor prognosis. In fact, the differentiated histology and this presentation that I showed you of the child with the kind of blueberry muffin presentation of tumors was called stage 4S. And this is a special presentation in which the tumors actually spontaneously regress, or the child's are treated with very little therapy. There's also this interesting finding of what they call neuroblastoma in situ. And this was the finding of rests of neuroblast in the adrenal glands of children who've been autopsied for reasons other than malignancy. And the frequency of finding these neuroblastomas in situ, higher frequency, like 1 in 500, than the frank presentation of the disease. So this suggests that you can have these growths occurring and then they spontaneously regress. But the thing that really fascinated me about neuroblastoma were the very early indications, of therapy-associated differentiation, in which and so when I came to neuroblastoma, it was part of small round blue cell tumors of childhood. We couldn't tell them apart from rhabdomyosarcomas or Ewing sarcomas. But with molecular genetics, which was really what brought me into the neuroblastoma field, people began to delineate genetic alterations in neuroblastoma. And of course, the most famous one for neuroblastoma was the finding that there was amplification of this gene related to CMYC called neuroblastoma MYC. And interestingly enough, this amplification of NMYC, let me go back, was associated with loss of chromosome heterozygosity at chromosome 1p. So here was the site of what we thought was the putative tumor suppressor gene. So you had an oncogene, a gain of an oncogene, and the loss of tumor suppressor genes. And you can see here in this panel that patient survival of children whose tumors have MYC amplification is really quite dismal. And this was, of course, the experiment that really fascinated me about neuroblastoma. So you could take these tumor cells from these high-risk patients that had all these genetic alterations, yet you could treat them with pharmacological doses of retinoic acid and control growth and induce differentiation. This suggested that the pathways to controlling growth and differentiation were not completely blocked, but that they were altered. And so with the identification of MYC, it was really the next step in which we began to ask what happened to NMYC. And so retinoic acid treated cells, you could see in this, this is like 1984, I'm sure it's before many of you were even born, where you can see that they have, this is called a dot blot, in which we put different amounts of RNA and diluted it, and then came back with a radioactive probe. And you can see the control have much more NMYC, while those that had the cells treated with retinoic acid had a marked decrease. And so we saw that this decrease in NMIC preceded the morphological differentiation of the cells. So this was sort of the first targeting agent for NMIC. This then, after many studies about retinoids, was then Kate Mathey and Pat Reynolds led a clinical trial in which they showed that after high-dose chemotherapy and bone marrow transplant and immunotherapy with anti-GD2, if you included retinoids, these patients had an improved survival benefit. We don't really know what's going on in the patients with the retinoic acid, so it still is a mystery. But in the United States, high-risk patients receive retinoids during just after their consolidation phase. And so I've been, studying what are, the mechanisms by which retinoids are effecting these changes, with the idea that if we understand this, maybe we can find some more targeted agents to help. And also, it came to be that we felt that these genetic alterations led to epigenetic dysregulation in what we call these neural crest precursors. And this contributed to the failure to differentiate. But the first thing was to try to understand what NMIC is doing. And so here you can see that when you silence MICN expression by treating neuroblastoma cells with a silencing siRNA, this results in decreased cell growth. So obviously NMIC is driving growth of the cells. And when you look at the morphology of the cells, you can see here that the cells that have received or have lost MICN function have a much more differentiated morphology by the extension of these very long neuritic processes and that are enumerated here on the side. So when we look at this, we can say that in a schema, ATRA can block NMIC, but NMIC is really blocking differentiation. So the overexpression the overexpression of NMIC is really suppressing differentiation, and it's stimulating growth. But what we didn't know was what was really the molecular mechanisms. And, as our ability to query cells has increased, when I studied initially as a postdoctoral scholar, we were looking at one gene at a time. Now we can look at all the genes at one time, which is really pretty remarkable. And that's why I'm so excited about the future for all of you young investigators, because it is a time when one can really get a feel for what's going on. So one of the things that's important to understand is that a cell receives a signal from the extracellular milieu. It gets processed at the cell membrane that activates different cellular pathways. And these cellular pathways, then the intracellular pathways, ultimately converge on regulators of what we call the epigenome. And I think the epigenome is incredibly fascinating because it is these alterations that don't occur to the actual DNA permanently, but influence the regulation of these genes. And it's what makes even two identical twins can have differences because their environmental signals can be processed differently. And we think very simplistically of the epigenome as a series of enzymatic activities that converge on proteins that can add modifications to DNA and histones called writers. And so this nomenclature is called, for EZH2, H3K27 trimethylation. And this trimethylation is a silencing mark. So it makes the DNA less accessible to polymerase to be expressed. Or you have ones in which you can erase these marks, or ones that interpret the marks. And so this is quite a combinatorial gives us many more combinatorial ways to regulate gene expression, because all the cell has to interpret all of these, and there's competition for different signaling molecules at the DNA level. So we were very interested in EZH2 because EZH2 is very important during development. And we went back to our neuroblastoma patients and began to look at the level of EZH2 in neuroblastoma. And we found that in ganglioneuroblastoma, you had very low levels of EZH2, and the same, relatively speaking, in ganglioneuroma. These are low to intermediate risk tumors. But the levels of EZH2 were quite high in neuroblastoma. Additionally, EZH2 resides on chromosome 7, and there were cases in which there were focal amplification of EZH2 or gains of chromosome 7 in neuroblastoma, which could lead to increases in EZH2. So we began to model, since we knew NMIC was regulating differentiation in this kind of big black box, we hypothesized that perhaps it was affecting EZH2. And so we have here a TET-off system in which when you remove tetracycline here, it allows expression of NMIC. And when you add tetracycline, you lose expression. And so we did this experiment in which we looked at how NMIC expression affected EZH2. Now, EZH2 is part of a complex called PRC2, in which the other components are SUS12 and EED. And you can see as NMIC increases over time, there's a gradual increase in EZH2, SUS12, and EED, as well as an increase in H3K27 trimethylation. And then when we looked at how does this affect a tumor suppressor gene, a gene called CASI1, we can see that NMIC increases, and when the EZH2 levels reach a certain level, you begin to see a decrease in this tumor suppressor gene. It's not only this one CASI1 gene that's lost, but there's a whole series of genes that are associated with differentiation. And so here is mouse embryo fibroblasts that have biallelic loss of EZH2. And you can see in those cells, they have very high expression of CASI1, TRAC-A, which is the tyrosine kinase receptor for nerve growth factor, another differentiation gene we're interested in, as well as one of the receptors important in retinoid use. And if you silence EZH2 in neuroblastoma cells, you can see that these genes are increased as well. So this suggests that NMIC is stimulating levels of EZH2, and then EZH2 is suppressing these differentiation genes. Oops. We also confirmed this using in a collaboration with Kim Stegmeyer's group that shows that the MICN amplified had a dependency on EZH2, and if you knock this out or decrease it with a pharmacological inhibitor of EZH2, then you could decrease neuroblastoma cell growth. So there is a drug an EZH2 inhibitor that's approved for epithelioid sarcoma. There was a pediatric trial that started with these ENI1 deleted tumors, which are rhabdoid tumors, in which there were some responses. As a single agent, we don't really think these kinds of things are going to be that effective. And so there aren't currently clinical trials with EZH2 inhibitors in neuroblastoma. But it is something that we, as we try to move the field to get forward in looking at combinations, it's really a drug I think that should be looked at again. So when I talk about transcriptional dysregulation occurring in neuroblastoma, and this is leading to alterations in differentiation, we have to go back and now to our neural crest model. And it's really important to understand that during development, lineage specification is really laid out by the differential expression of transcription factors. And so you can see that in this cartoon, the migrating neural crest may have expression of these transcription factors. And then as they develop, they gain expression of other kinds of these lineage specifying genes. And these are very important in their both their specification as well as what they're going to be when they finally grow up. And it's this set of transcription factors that are very important in normal sympathoadrenal cell lineage specification and differentiation. And it's thought then that in this model, the gain of NMIC is really contributing somehow to neuroblastoma. So if you think of neuroblastoma in a developmental context, it really is a cell that has is in a constant self-renewal state. And it's driven and dependent on these neural crest cells. In a really important paper that came out a few years ago now, these lineage specifiers form an integrated network. And this network is really both, is a feed-forward network in which the different members of this transcription factors that specify neural crest developmental programs both collaborate and stimulate their own production. And what Adam Durbin found was that the MYC-N gene was like an input into this core regulatory circuit. And because it was there, it was creating a dependency on the neuroblastoma tumor cells on these particular transcription factors. So that if you inhibited the expression of these transcription factors, then they had decreased growth. So we could think then that if NMIC is stimulating, in this model, we showed EZH2 is inhibiting differentiation genes, and it's inhibiting the tumor suppressor genes. And that's the break, okay? And then NMIC is stimulating these lineage-specific transcription factors, which are impacting growth genes and self-renewal. So this is part of the model. So what I was interested in understanding, because I came from a time in cancer biology in which, in a simplistic model, tumor cells were driven by the gain of oncogenic events that stimulated growth and the loss of genes that prevented growth. So we wanted to ask, what was Casi-1 doing in this model system? And so we developed TET regulatable systems, and now we have a TET-on system where when you add tetracycline, Casi-1 expression goes up. And you can see very nicely that in the control cells, they grow, but when you turn on, treat the cells with tetracycline and Casi-1 is expressed, then these cells have decreased growth, okay? And you can see in this picture that these cells with Casi-1 expression have very long neurites. So, whoops, sorry. Have very long neurites. So how does the loss of the tumor suppressor, Casi-1, affect this circuitry? Does it impact on the core regulatory growth circuitry? So It wasn't easy, but we were able to develop some systems so that we could look to see where Casi1 transcription factor binds to the DNA. And using chip analysis, this chromatin immunoprecipitation analysis, and our model system where we could add tetracycline and increase expression of Casi1, you can see that and I'll walk you through this one a little bit slowly, as there'll be others. You can see that these peaks are increased Casi1. There's a basal level, but then when you add Casi1, you get increased. And here you can see over this particular gene, H3K27 is considered an activation mark, and genes are usually expressed. But now when you increase expression of Casi1, the activation mark goes down, and you also have a decrease in binding in RNA polymerase II. And when you look at the number of reads of RNA that you see over this GATA2 gene, you can see a marked decrease in expression of GATA2. And the same can be seen over here for another one of the core regulatory circuits, TBX. So basically, the presence of Casi1 is actually a brake on the expression of this transcription factor. And if we look more globally, you can see that actually all the core regulatory circuitries are inhibited to varying degrees when we restore expression of Casi1 in neuroblastoma cells. So this says that Casi1 acts as it binds to predominantly these enhancers, and then inhibits the expression of this core regulatory circuitry. And this we recently published in Cell Death and Disease. And so I think it really very nicely shows that the loss of the tumor suppressor can also have a role in amplifying the growth signal by releasing a negative regulatory element. So Casi1 forms a negative regulatory component to the CRC. So this led to us to reconsider now, if we think about our little model or cartoon in this Waddington plot of the differentiation of a sympathoadrenal, and now we have a neuroblastoma cell and we treat with retinoic acid and we can differentiate them. What is the core regulatory circuitry involved in determining this neuroblastoma differentiation state? And to do this, we utilize some similar kinds of technologies that Adam Durbin and others had done to identify the core regulatory circuitry that was involved in neuroblastoma self-renewal. And so we wanted to identify the master transcription regulatory factors that occurred during neuroblastoma differentiation. And this was done by a postdoc in my lab, Deblina Banerjee, and a postdoc at the time, Berkeley Greider, in Javed Khan's lab, who also runs his own laboratory now, and an MD PhD student. And to do this, we analyzed H3K27 acetylation ChIP-seq at various times after neuroblastomas were treated with retinoic acid. And then we identified these structures called super enhancers, which were these really large pile-up or reads of H3K27 acetylation marks over particular transcription factors. And what we saw was there was really a dynamic reorganization of super enhancers. So here are the cells that are treated for at the control cells at 2 days, at 4 days, and at 8 days after differentiation. And now here are the H3K27, these are called hockey plots. So the ones that have the highest signal, and this one here for MICN is way away from all the others. But you can see here the HAN2, GATA2, TBX3, FOX2B, all of these, which were actually, part of the core regulatory circuitry. And this core regulatory circuitry kind of changed dramatically as you treat with retinoic acid. And we used a set of, and you can see the top ones change with time, okay? This is very interesting here that MICN comes back again. And what I have to say about this is the fact that these are all RNA-based assays, and it's looking at the transcriptome. And what happens with retinoic acid-induced differentiation is that initially there is a RNA-based decrease in MICN transcription, and then there's the upregulation of a proteasome-based degradation of MICN protein. So in point of fact, there's no MIC anymore protein in the cell, but the levels of MICN return again. But you can see a series of different factors coming into play as the cells differentiate. And we then looked at the signals, okay, and here you can see that MICN is going down. We saw an increase of SOX4 and SOX11 and GATA2 and GATA3. And these were quite interesting to us because some of these genes are differentially expressed during normal neural crest or sympathoadrenal differentiation. So we then used, we took the whole set of these super enhancers that we had identified, and we used k-means clustering to identify populations that were sort of most analogous to one another. So the minimum number of groups that the data could be clustered into with confidence. And so we found what we called the first wave, a second wave, a third wave, and then the lost super enhancers. And it's probably a little easier to be seen this way. So this is the H3K27 acetylation score for a group of genes, okay, that were very high in the control cells, but with time were lost. Then we had a set of genes that were, relatively low, but then were gained transiently. So we call this the first super enhancer wave. And then we had another one, which was the second, a group of genes that came on much later. And finally, a third wave that seemed to come up at the very end. When we looked at what these genes were, so this is the nice thing by analyzing all the genes possible, we can then, in an unbiased way, look and see what kinds of groups do these genes look towards. And in fact, the genes that are lost in the lost cluster are stem cell differentiation and development. And the first wave are those that are negative regulators of signal transduction and communication. And then the third are the regulation of neuron projections and apoptosis. So in some contexts, it makes a lot of sense. So initially, you lose those genes that are supporting the stem cell component of neuroblastoma. And then there's a whole series of genes that come up that are involved in, just broadly put, regulation, okay, in very indistinct ways. And then the second wave where you're more getting into the differentiation, which kind of makes a little bit more sense. So to identify the ones in this retinoid, the master transcription factors. So these are just where we see H3K27 acetylation peaks, okay? And then you have to correlate the peaks with the genes. And this is a bioinformatic tool. We did spend a lot of time validating several of these where we went on to do further biological studies, okay? So, and we wanted to filter them. So first we identified the super enhancers and then looked to see what the target gene was associated with them bioinformatically. And then we took that list and filtered it for transcription factors. And then we asked the question if the super enhancers were correlated with what we saw for the RNA-seq signals, meaning when we lost the RNA, the super enhancer, was that associated with a decrease in RNA? And if you gained, super enhancer, was that associated with a gain in RNA? And so from that, we then began to parse out the loss cluster and the first and second wave, okay, in which, based on those criteria, we had 13 super enhancers in the loss cluster, 10 in the first wave, and 18. And I think on the next slide, this shows how the transcription factor expression also changes. And these are the actual genes and the heat maps to show. So you can see, for the most part, there were 8 transcription factors here. And luckily, MICN was one of these. And then in the first wave, you see these series of genes that are transiently expressed, SOX4, PPAR gamma, and then a whole series of genes whose expression is more stabilized. So these were components of the CRC and MICN, so that was very satisfying. So then it was a real challenge to figure out, of all these genes, which ones to go further into to determine some of their activities, their biological activities, because this was a lot of bioinformatic, data analysis done by Berkeley Grider. And I'm a biologist at heart. So, from this list, how are we going to determine which ones are we going to go off and do the hard biology on? And so he did this linked enhancer activated leaf plot, which really tried to get an idea of those that would be the most important in analyzing. And so in the control cells, SOX11 came up as a really important one, and SOX4 came up in the at 2 days and 4 days after treatment. And so these are the ones that we decided to move forward with. And so the first thing we had to do was to make sure that the ATRA led to a loss in the super enhancer. And so here you can see, these are the plots of the SOX11 gene here. And this is the super enhancer, which is distal, is distal to the gene. And here it's blown up. And you can see quite nicely that the SOX11 super enhancer is lost as we treat with retinoic acid. We also confirmed this making CRISPR-guided to targeted ones to show that when we transfected the cells with these silencing CRISPR guides, we could see decreases in SOX11 to really confirm that they were controlling SOX11 expression. And you can see that when we decrease these super enhancers, you have a decrease in SOX11 mRNA levels. So this was a confirmation of the bioinformatic approach that we had used to link these super enhancers to this particular gene. We then asked the question, what happens with the inhibition of SOX11? And so we used siRNAs and we decreased SOX11. And you can see with the siRNAs, to varying degrees, we were able to inhibit the growth of these cells. And here you can see over time, and the morphology of the cells. We also made a short hairpin SOX11 cell and injected it into animals. The controls you can see here, and the tumors with the two different short hairpins, you can see there's decreased cell growth by weight in these tumors. So not only is it affecting the growth of the cells in vitro, but also decreases neuroblastoma growth in vivo. And so the question was, how does SOX11 affect the neuroblastoma core regulatory circuit? And in point of fact, if you delete SOX11, it decreases the neuroblastoma CRC. So, so what's happening is that this is another gene that's actually input into into the CRC. And in fact, it does affect MYC levels as well. Some of the transcription factors are not affected, while others are, and there's a lot of heterogeneity in this response. This is mRNA levels, and this occurs also at the protein level. So we feel that SOX11 functions as an input into the neuroblastoma CRC, because when you silence it, then if you silence all of the members of the CRC, you have very little to no change in SOX11. So here's how we would diagram this particular, that SOX11 functions as an input into the core regulatory circuitry. And SOX4 induces, again, in the SOX4 super enhancer. So here we have increases in there's a super enhancer here, which is quite distal from the promoter region, and then one that's more in the first intron. You can see that there are a big increase in the super enhancer here early on. And we also knock this down to show that this was also linked to the particular SOX4 gene as well. And you can see that, the relative SOX4 mRNA expression, when you use the different guides, is attenuated. So when you inhibit this, you have a loss. So we feel these super enhancers are responsive to retinoic acid and affecting the growth. And the loss of SOX4 expression attenuates ATRA-induced differentiation. And so you can see here increases in tubulin, DPYSL3, and SOX4, and increases in neurite length. And then finally, we looked at neuroblastoma patients, and those that have low SOX11 and high SOX4 have amongst the best prognosis. So in our model for differentiation, the sort of circuitry required is a shift from a self-renewal state to a differentiation state that's really influenced by these transcription factors. Of course, big questions are in the future, what do the differentiated neuroblastoma actually become? And so we're doing a lot of work with a neurocrest biologist, Laura Caruso-Pahlberg, and we're working with bioinformatic people in analyzing single cell data to truly get a handle on, are they taking a differentiation pathway that's a normal differentiation pathway, or is this a hybrid differentiation pathway? So I hope today I've shown you that we've begun to fill in some of the black box in how NMIC leads to dysregulation of neuroblastoma, the role of ATRA in modulating the pathways that lead to the dysregulation of neuroblastoma growth and differentiation. I think there's still a lot to be done, because even though we've worked at it, in a lot, this seems like a lot of details, there's still a lot that we don't really know. And one of the big efforts ongoing in the lab by Jui Liu, who's the senior associate scientist in my group, is trying really to understand how does NMIC interact with these different molecules to affect its transcriptional changes, and are the co-regulatory proteins that it recruits to the transcription sites of transcription to recruit RNA polymerase, how are they involved? And will this information lead to new insights that can lead to perhaps therapies that we can add to the current ones? Here, he's done an initial immunoprecipitation looking for interactors of NMIC, and it's really quite remarkable. There are over 340 common proteins. They come in these different classes. And here you can see that NMIC is interacting and binding to complexes that contain the neuroblastoma core regulatory circuitry. Sorry, seems to be on auto forward. And it also is interacting with complexes important in chromatin remodeling. And this is another area that's really important that we're investigating as well. And so these studies are ongoing and amongst the most interesting in our future directions. So I'd just like to end and thank, I've been really blessed to have a great group of people. Jui Liu, as I mentioned, leads my laboratory. We had a terrific postdoc, Manzu, who did a lot of this work. We have a whole effort in immunotherapy that I haven't had a chance to talk to you, and neuroblastoma heterogeneity that was led by Ming Sun, as well as a lot of young people who come to the lab for a short period of time, and their contributions are really terrific. Of course, we're really lucky to have a lot of core facilities that are supported by the Center for Cancer Research at the National Cancer Institute. And I have terrific collaborations with my pediatric oncology branch co-members, as well as other laboratories, specifically Javed Khan, who's had a long-term interest in neuroblastoma. And so with that, I'd be happy to take questions.
Thanks a lot, Carol. We're sending you online applauses. Your work is always outstanding and fascinating. The four groups, the four waves of super enhancers is just beautiful. I have a few questions. We have, let's look at the chat. Professor Manuela Martins is congratulating you. Excellent talk, and thanks. So it's beautiful to see how over the years, like groups like your group are moving from looking at very specific hypotheses and very specific systems towards larger amounts of data and analyzing larger amounts of molecules at the same time, and gene groups and so on, because of all the new tools we have right now. So we have a question from Barbara, our student. I don't know if you can read. Can you read it there, or do you want me to read it out loud? Oh,
yeah, I can't
read it anyway.
I can't see the questions. Yeah.
Yeah, I should read it anyway. So Dr. Barbara Kunzler is asking, who's a former PhD student co-supervised by myself and Dr. Carol Thiel, is asking, do you think it's possible that another epigenetic repressor like J9A, for example, since it's overexpressed in neuroblastoma, is compensating for the fact of EZH2 inhibition? And do you consider combining dual inhibition of these targets?
Yeah, hi Barbara. Yeah, that's a great question. We do have a lot of data on the NMIC interactors that are regulating the differentiation and the self-renewal. And I do think that those combinations will be really important, okay? If you think about NMIC as having, it's putting the accelerator on and it's relieving the brake. And if you can take the accelerator off and put the brake on, then I think you can get at it. And so, we hope to have it published soon, but it looks like definitely combinations, because there's two different classes, and there's been a lot of controversy in the MYC field because some people believe that MYC is only a transcriptional amplifier, and that any negative regulation that occurs via MYC is indirect. And we actually have evidence that MYC-N is binding and negatively regulating. And so it may be that NMIC may be different than CMYC. Oh, okay, I can see the questions out. Yeah, and definitely G9A is involved in the repression of the differentiation genes. So I definitely think that this is going to be something that's going to be important. But we also have to relieve the activation mark, and that we believe is being mediated by CAT2A. So we hope to have that published soon.
Okay. Professor Saulo Andrade from our School of Pharmacy, and congratulating you, nice presentation. Professor Marco Stefani is a neurosurgeon and neuro-oncologist, also professor of, here at the university, and they're both collaborators in our grant. Congratulations, that was a fascinating presentation. And Professor Guido Lenz, who works on cancer biology at our Department of Biophysics and Center for Biotechnology. Great work, congrats. Is the sequential expression of genes observed in populations also observed in single cells?
Yeah, that's a great question. So we haven't done this experiment in single cell. It seems like the next logical thing, which we should do. We are a little bit distracted in the laboratory by this concept in neuroblastoma that I didn't really mention, but the phenotypic switching that goes on, the tumor cell heterogeneity. So we spent a lot of time working on that. This is a project that Ming's son has developed on, trying to understand what's controlling the phenotypic switching that occurs. And so I want to do the retinoic acid single cell experiment because I definitely think that there are different populations. And if we parse that out, it'll be easier. Single cell is really good for some things, but it's very, I think we'll have to do single cell ATAC-seq will be the best way to approach that question, because the ATAC-seq signals are more robust than the RNA-seq signals in single cell analyses so far. You don't get a lot of lowly expressed genes in the single cell.
Okay.
But it's a great question, yeah.
He's a very smart guy. Giulia Vanini is a PhD student in our lab. Congratulations for your amazing work. You showed track A at some point as relevant, and you've been working on neurotrophins and track receptors. You've been working a lot in the past in neuroblastoma. And Do you think there's a link between, there's a nexus between among track signaling, MYC, and epigenetic mechanisms? Because you showed in a slide that activation of growth factor receptors and PI3K and all the kinases eventually leads to changes in epigenetic regulation. So what's the link between track receptors and the epigenetic?
Yeah, so that's a great question. We kind of moved away from the track area, and it is a natural. We know a lot about the signaling, the growth survival signaling that tracks are mediating, and I still think it's so important. It's hard to get the clinical people to use these kinds of drugs up front with chemotherapy, although they are using. I think the latest ones here are going to use an ALK inhibitor up front with chemotherapy. I still think some of the other ones might be, better, like the PI3 kinase or an AKT one.
Mhm.
There's a lot of people who still have this idea that they're influenced by the old way of thinking is that more is better, not less is better, and it's hard to get them to recalibrate in that context. But we haven't really pursued tracks in the context of understanding epigenetically what happens. I mean, it is interesting that EZH2 is definitely suppressing track A, and it definitely comes up. And the cells are more responsive to NGF when they are, when that goes away. Which, I think this makes sense when you think about developmental. So if, in point of fact, if during development NMYC is stimulating EZH2, that keeps the levels of track A down, then when they get to that point in development of other tissues that are exporting NGF, then they're either not going to be there or they're not going to differentiate, or, so that's why I think pediatric tumors are, the developmental aspects influence them, and it should, especially in neuroblastoma, give us an opportunity. I would like to put a shout out for everyone. In 2025, the neuroblastoma meeting is going to be here in Washington, D.C. We're hosting. The Mid-Atlantic Consortium is hosting it, so we'd be really excited to have people come.
Oh, we can do that, definitely. Let's plan that. So yeah, but there's a shift during neurocrest development in NGF track A signaling, right? Which goes on in parallel, more or less in parallel with the epigenetic shift from lots of proliferation to differentiation. Very interesting. Professor, yeah, go on.
No, I was just going to say there's a whole, we don't really know. There's a big lesion in our understanding, all of these models that we put together from neuroblastoma cells are, we don't really know what's going on in normal neural crest cells. So, that's why it's really important. I've been working with this young investigator, Laura Caruso-Palberg, who is a neurocrest biologist, and she's making, human trunk neural crest.
Mm-hmm.
So hopefully we can ask these kinds of questions to see how related the genes are in neuroblastoma to what's going on in normal neural crest. Because I have to tell you, she had a hypothesis. I mean, NMYC is known to be a neural gene, a neural progenitor gene, and a lot of its studies has been in the CNS. And Laura found NMYC expression in neural tube cells, and has a paper in PNAS several years ago showing that it's possible that these NMYC, neural tube cells get caught up in the stream of migrating neural crest cells, because she doesn't see a lot of NMYC expression in true neural crest cells. So I always like the out of the box and different hypotheses. I mean, and I think all bets are off until the data is really solid.
Yeah.
You need to be flexible. When I started studying, molecular biology, it was one gene, one mRNA, one polypeptide chain. That was the central dogma of molecular biology.
Yeah.
Well, that is clearly not true, so.
Yeah, it's much more complex than that. And I agree, we have to look at the normal embryo to understand pediatric cancer, right? Increasingly.
Yeah.
So Professor Saulo Andrade, he's a drug developer here, professor in our School of Pharmacy. In your experience with NMIC, what are the most promising pharmacological targets to decrease its expression in neuroblastoma beyond aurora kinase? Is there another kinase that are promising targets?
Well, so there's an Italian group that has this OncoMIC, and it's kind of a decoy MIC drug, and they are getting some traction with it.
Mm-hmm.
We've as part of Jui's story with NMIC and understanding the coactivators, we know that CAT2A is involved in degrading NMIC. And so I think these degrading strategies are getting more and more clever. And so I do think it's going to be possible that we may be able to do targeted degradation of NMIC in the future. So I think that's a possibility. And as I said, we tried to make a CAT2A degrader because we thought that the CAT2A was affecting the stability of NMIC. And so maybe if we could get rid of it, we could decrease NMIC stability. So.
Okay, we have more questions. Our collaborator, Dr. Karina Munoz, is congratulating you. Great work. Thank you for sharing. Saulo Andrade, great talk, Dr. Carol. Thank you. In terms of heterogeneity, are there any possibility that these epigenetic treatments are selecting specific groups of cells that can turn resistant or recurrent?
Yeah, no, I definitely think that's true. We've known for a long time with retinoic acid, prolonged treatment with retinoic acid, we end up getting retinoic acid resistant cell lines. And we published a paper a long time ago that got kind of hidden in the first issue of Cell Death and Differentiation, that, you can select for a cell that makes a lot of IGF-2, and this gives it a survival advantage against the retinoic acid. And they believe that some neuroblastoma mesenchymal cell types use retinoic acid as a growth factor. We haven't necessarily seen it in some of the cells. We've recently made a whole series of adrenergic and mesenchymal cells from patient-derived xenografts. So they're not well-established cell lines, and we haven't necessarily seen that retinoic acid is stimulating the growth of our mesenchymal type cells. So, but I definitely think that, cancer cells are smart, and because neuroblastoma especially is, an embryonal tumor, many of its pathways are, we used to call it a bivalent state. It's developmentally poised to do a lot of different things. And so I do think that's a challenge. And if we can understand the nature of what enables it to be poised, then I think that will give us a clue maybe to decreasing heterogeneity.
Yeah. Do you believe neuroblastoma can share a biological origin with Ewing sarcoma? Because your friend Elizabeth Lawler has shown that a Ewing sarcoma can arise from neurocrest cells as well. That's one of the hypotheses. I believe this is a strong hypothesis. And I think I remember your review paper on epigenetics with Elizabeth Lawler, and you were talking about epigenetic. You were talking about neuroblastoma and Ewing sarcoma in parallel, if I remember correctly.
Mhm. Well, so it's interesting. I think that, the origins of Ewing sarcoma is really interesting. I think as we are, as the single cell atlases for different tissues are evolving and people are understanding better the plasticity. I mean, I do think mesenchymal cells contribute to, the disease. Some of the best kind of in vitro models are for Ewing's are mesenchymal in origin, and I think it's tricky. Same with neuroblastoma. I mean, you have mesenchymal types, and uh, there's even I can't remember the old data, but Bernie Weissman used to do these things by putting different things into Ewing cells, and I mean, Ewing cells will express markers that have classically been considered neural phenotype. I don't know whether that's a prejudice because nerve growth factor and its name may prejudice people to think it's neuronal, but where it may not be. But the Ewing sarcoma cells are cholinergic, so they do make neurotransmitters. Um, they have
And
high levels of
they're prone to neural differentiation too.
yeah. So I do think that it's. I remember the first time we put a paper in, and they said everybody knows it was implicating the neural origin of a neural origin of, or positing a neural origin for Ewing's, and they said everybody. Then one of the reviewers said, "Oh, everybody knows it's a bone tumor."
Yeah.
Well, I haven't seen it
We
make
know it isn't.
osteoclast or stuff like that yet, calcified bones. So I think it's more complicated than that. So there's a lot of interest in different types of mesenchymal cells. Just like, now there's lots of different types of fibroblasts. And
For a young researcher in the field of childhood cancer biology and pediatric oncology, what would you say is the future of the field, where it's heading in terms either of what are the major current challenges and future challenges, or what are the most promising approaches technically, or where are we heading in terms of biology of childhood cancer?
well, I think biologists have to move into, excuse me, they have to become computationally savvy and learn how to use large data sets.
I know
That's
I'm
the
too old
first
for
thing.
that, but definitely, yeah.
Yeah, or they need to have really good friends who are
Yeah, that's right.
data people, because this is just the wave of the future, and it enables you to ask questions, that are both intuitive but also unbiased in some ways. And the fields that I think are really fascinating, I think the cell-free DNA field is extremely fascinating because what they're being able to discern from a blood test, in terms of the epigenetic status of the cells, is really quite remarkable. Also, I've heard that you can detect where the tumor may be, because if it's causing tissue damage in a particular tissue, each particular tissue has a type of DNA that it's shedding. And so I think this is a wide open area, um, and certainly for our patients, it's going to be great. It's not going to be so great for us who want tumor specimens to sample, because I don't think we're going to get second look biopsies, but I think that's an area. Uh, I mean, I think the intersection, I started as a tumor person,
Mhm.
and now you have to really look at what the tumor is doing to the microenvironment,
Yeah.
and then how the microenvironment, what that's doing to the tumor.
Definitely.
And of course, the immune system is really hot, so immunologists are in vogue again. So I think that this, intersection of these disciplines is really where things are happening. And it's hard for one person to be everything. So that's where you really need to have good teams. And I think the leaders need to be much more. Well, the analogy I like to, hold, water is the life force for everything. You can't hold water in your hands if you grab everything too tightly. You have to hold it loosely. So we need to work with each other, in a very open and, integrated way in order to move things forward.
Yeah.
Because it's just too hard to do these experiments.
Yeah.
I looked at my first paper, and there were like 4 figures. And only one had an insert. That was pretty unusual.
I was just rereading that paper the other day, and I realized that, and I miss those days. Even in the 90s, I think it was much more romantic that you could get a very innovative breakthrough Nature paper with 2 figures, and now you need the 36 supplementary figures just to publish in whatever journal. So
Well, and that's it's like I get tired reading the papers. Who would want to read the papers? No wonder they have these briefs, to tell you what the paper says. But, that's kind of sad because when I've gone back and looked at some of the papers, I'm even surprised at some of the things, I said, oh my God, that was pretty insightful,
yeah. I think
considering what I know now and what I knew we didn't know then, it's pretty amazing. So
things are not straightforward anymore in papers
yeah.
like. Well, this has been fantastic, Carol. Thanks again. Thank you so very much. We had a peak audience of around 50 people, I guess. I guess it's pretty good for our online seminars. So
Thank you. It was great.
So very much. Thank you so very much. This has been a great talk and great discussion. It was a pleasure and an honor to have you here. Hope to keep counting on you to support us as you've been doing for.
Yeah, definitely. No, it's been fun for me.
Thank you so much. And so, hello. Oi.
You have an assistant.
Yeah, this is my assistant. Yeah, I would like to thank you as well, Caroline. And
Hold
actually,
on. I want to thank the Felipe yourself. I want to thank you and the Center for Biotechnology for providing this space, one of your seminars, for providing the seminars to our group to promote that. So thank you so very much.
oh, that was a big pleasure, and I think we should have more and more of those joint efforts to cover a lot of topics in the seminars, and have people like Carol with us. It's a big pleasure, really. So thank you very much for taking the time for that.
Yeah,
and we have 117 visualizations of the seminar already. So that's a lot.
Oh, great! Thank you very much.
Pretty good.
Okay. Thanks so much. Yeah, Raphael, thank you, and we'll keep in touch, and I'll be asking you for a letter because I'm going to be reviewed in next May. So.
Oh yeah, definitely. Sure.
Yeah.
Sure. And this talk will be available online on YouTube.
Okay.
So I'll send you the link.
That's scary.
Yeah, no, I know. But I mean, yeah, it's okay. It was beautiful.
No, I mean, it's not scary from a data point of view. It's scary just listening to myself. That's always scary.
Just too shy. So thanks again. Have a great weekend, everyone. And see you next time.
Bye.
Bye-bye. Have a good weekend.
Bye.